Evening, everyone. Good morning. I'm Nikita Thakur, Department of Chemistry, speaking here today at National Level Online Faculty Development Program, organized by Sardar Patel College of Pharmacy, and association with GTU on the topic Smart Research for the Digital Age Master and Grant Proposal. High Impact Scope is publishing patents and AI-driven excellence. On behalf of SPCP, would like to present a very warm welcome to the respected faculty, esteemed guests, and our fellow participants. I welcome you all to the third day of our FDP. Yesterday's session set the stage for enriching experience. It was wonderful having Dr. Vandana Patrawale ma'am, and ma'am has effectively outlined unlocking the mysteries of research grant writing with meticulous attention to the detail. And today we promises to be equally insightful. Let's dive into the discussions, learn from each other, and take back valuable knowledge to our institutions. Today, we have an exciting lineup of sessions designed to enhance your research, review, and industry engagement skills. By a profound speaker, Dr. Ketan Ranji, sir. Let's build on the momentum from yesterday and make the most of this opportunity to learn, share, and network with fellow educators. So now I would like to welcome Ms. Gunjan Rimani, ma'am, for the introductory speech.
Good morning, everyone. Am I audible? Am I audible to everyone?
Yes, ma'am. Yes.
Okay. Good morning, everyone, to all the participants. Good morning, Ketan sir. Thank you, Nikita ma'am, for giving me this opportunity. It gives me a great pleasure to introduce our esteemed speaker for today's session of the Faculty Development Program. I request all the participants, please join me in welcoming Dr. Ketan M. Run sir. Dr. Ketan sir have completed M.Pharm and PhD in Department of Pharmaceutics. In an accomplished academician and researcher serving as associate professor and head of the Department of Pharmaceutics at LM Pharmacy College, Ahmedabad, Gujarat. With a distinguished academic background, Dr. Run sir obtained his B.Pharm with university honors from Sardar Patel University, Anand, an M.Pharm from NVP College of Pharmacy, Pune University, and a PhD from LM Pharmacy College, Gujarat. Dr. Run sir brings over 20 years of experience in teaching, training, and research in pharmaceutical sciences, complemented by a year in industrial research at Ranbaxy Research Labs, Gurgaon. His expertise spans formulation development, focusing on novel drug delivery systems, ocular drug delivery systems, leveraging nanotechnology. He has authored over 50 research publications, from which many journals have impact factor more than 11. Significant a recipient of multiple research grants, including substantial funding from DST, Gujarat Coast, international collaboration in setting research funding exceeding over more than rupees 2 crores, which includes funding from DST, FIST, GSBTM, Coast, DST Stuti, and also from industrial projects like Leolens Tech from USA. He has also undertaken uh, consultancy and industrial projects contributing to innovations like herbal tablets and surgical gadgets commercially available through startups. Recognized as a Tech Guru Award for the Best Teacher in Pharmacy by GTU, Dr. Ketan Sir is a certified trainer for design expert software and conducted numerous workshops and training sessions across India and from also abroad. He had guided several PhD and MPharm students and actively contributed to the accreditation and organization of national and international conferences. Dr. Ketan runs a commitment to advancing pharmaceutical education and research is underscored by his leadership at LM College of Pharmacy, where he continues to shape the future of pharmaceutics through pioneering research and mentorship. Today, we are honored to have him with us today to share insights on the topic writing an effective research paper, which I am sure will be highly beneficial and enriching for all of us. Thank you, Ketan sir, for joining us today and for this wonderful topic. Without further disturbance and ado, let us welcome Dr. Ketan Ran sir to take the session forward.
Am I
Yes, sir.
am I audible?
Yes, sir.
Are my slides visible?
Yes, sir. Yes, sir. It is visible.
Okay, okay. Thank you, Gunjan madam, for a kind introduction. I am really impressed with your words. Okay, so at the outset, firstly, I am thankful to the Sardar Patel College of Pharmacy, the organizers, Principal Dhaval sir, as well as the coordinator Samres Pal Roy sir, and other faculty members of Sardar Patel College of Pharmacy for inviting me over here on this platform to share and learn something from what we have gathered in our past 2 decades of experience in academic research or in academic pharmaceutical. At the outset, I would also like to congratulate and appreciate the efforts and the contribution which the faculty members of Sardar Patel have put in to organize an online faculty development program on a theme which is very relevant, on a theme which is need of the hour, on a theme which would be something which everyone aspire excel in their career. So you have rightly notched up on something where every faculty member or every research scholar is looking to upskill themselves. And I feel really honored and privileged to be among the galaxy of speakers who have been my teacher directly or indirectly, where I was witnessing and I was as a participant in Maurya sir lecture and also in a part of Vanna madam lecture, where they have set the right tone as the anchor was briefing us at the start of the today's session. So to take it on to further, the topic given to me from the organizer is writing an effective research paper. Am I audible completely?
Yes,
Hello?
yes,
Yes,
yes, sir.
Yeah,
sir.
yeah.
Yes, sir.
So to just keep a very loud and a clear voice, I would turn off my video so to have a better networking between us. But before that, let me tell you about the at end of the session, we would come up with certain learnings and hopefully those learnings which you will be able to implement or execute at your workplace. Now, I am not the right authority to talk on this topic, but with little bit what I have gathered in my career with the support and contribution and efforts with my students and with my colleagues whom I have worked in past 20 years, I could publish around 40 research papers in good research journals ranging from International Journal of Pharmaceutics, Journal of Controlled Release, APS PalmSci or ACS Nano or Acta Biomateriala, with ranging of publishers from all Taylor and Francis to elsewhere to even MDPI and many more. And whereby I have been also engaging myself in reviewing the papers from different publishers. So on this little bit of experience, I will try to share something what I have. I'm also here to learn from you all, so that we all together can collaboratively and collectively make a better publications. So the session, I have kept a very specific and achievable learning outcome so that we would be able to understand, comprehend the structure of the original research article. I'll just take you through the different articles that are being submitted to the journal. And we will also see steps to structure a science paper, which editors will take serious note of you. So the first part, the major part of around 40 to 60 minutes will be on understanding and comprehending the structure of original research article along with the paper, my own paper, which is being published. And thereby, after understanding the different sections and different parts of the original research article, I'll try to guide you on how you can structure your research work, which the paper editor will take serious note of it. So on this particular note, we start our today's learning session to understand the structure of original research article, which most of the publishers takes into consideration is introduction, material methods, results and discussion, IMRAD. So this is something which we will be trying to discuss. Am I audible completely? Hello?
Yes, sir,
Some
you're
restaurant
audible.
or some organization. Okay, fine. So
Sir,
to
you
begin
can turn
with,
on
I
your
would
camera,
say
sir.
every
So you
yeah,
can turn on your
I'll
camera.
just I'll just turn off and turn on my camera based upon the network or the voice lag.
Okay, okay,
Okay.
okay.
I
Okay,
hope
sir.
my slides are visible. My slides are
Yes, sir.
visible.
Perfect, perfect, sir.
Yeah. Okay. So every one of us, being a teacher, researcher, or a scholar, has been face to face with a blank page at some stage of our career, whereby we are wondering where to start and what to write first. And to be very honest, describing one's own research work in a format that is understandable, that is comprehensible to the others, as well as acceptable to the publication, is not an easy task. So organizers have set the right topic whereby we all can be guided together to come down to a format of our research work which can be comprehensible, understandable, and acceptable for publication. All of us will also accept that we are investing lot of time, energy, and often money in our research, and we become intimately emotionally attached towards our research. So we as ourselves, we are very convinced of the value of our research, what we have undertaken for post graduation, for our doctorate studies, or for supervision of our students. And we feel that it is also of importance to our scientific community. But how do we communicate and disseminate this entire content to the scientific community is something through the publications. And when we try to publish it should be presented into a research which is here in understandable fashion, not only to the experts of the field, but even I expect personally to the non-experts in our field can also appreciate the interest of our findings that we have taken. So, as we were on the first day with Maurya sir lecture with an old age saving of publish or perish, which remains equally valid in even today's time. Okay, so many young researchers and not even young researchers, all of us find ourselves under pressure to produce scientific publications. This is in turn to enhance our career perspectives. This is in turn to substantiate the request for the research funding from different agencies, or maybe sometime to justify previous funding allocations that has been made to us as a part of completion of a grant, or as a university requirement qualification. Like many of the universities would also have a requirement of a scientific paper to be published, like GTU has its own requirement of publication of a paper with PhD degree, and so on. The other universities of India would also have. So we do require time to time up-to-date training on this particular area, and whereby I also feel it is not only a science, but it is an art of writing a scientific article. That particular art you will acquire on and only once you get involved and engaged in writing the manuscript. Okay, as the saying goes on, you can't learn the driving a car by reading a book of a driving or by attending a seminar and webinar of how to drive a car or how to swim in a river or whatever different aspects. These are some of the skills which you have to learn by doing. So what I personally feel is that you yourself have to engage in manuscript writing or in writing some sections or some part of the manuscript which we will go through across the presentations. So with this in mind, I will try to present a step-by-step guide to writing a research scientific article, which may not be a specific to the discipline of pharmaceutics, but maybe rather be applied to the vast majority of the pharmaceutical science discipline, medical discipline, or a paramedical discipline. And I'll try to outline the main sections of the article, and which will then be described in a greater detail with different elements, and that should feature in each of the section. So at the end, we will try to see that you create a good quality first draft of your work, which can be circulated among your team members, among your co-authors and senior mentors for further refinement. So this is something I expect as a written gift from you all after you complete this particular FDP. Now, apart from this setting, a stone over here is something what I feel is that it is also unethical to conduct a study and to report the findings. Okay, so this is something highly unethical, whereby you have some study and you are not informing to the scientific community. So maybe some of the other people are all doing the same work and wasting the time and resources. We also have some results that are worth reporting that will lead to the major other discoveries and other inventions. We want to also publish to give the credibility to all our co-authors, all our research scholars who are in our team. We want to reach to a broader audience, okay? Maybe to Europe, maybe to Australia, maybe to America, to the people, to the scientists who have been working in our area. And at the end, this is going to give credibility to our reputation. This is going to enhance the chances of promotion and advancement in our career. And we would also like to obtain more research grant, and that is only possible when you have some number of papers in your name. Okay, so let us convince ourselves, yes, we should do the publications with utmost care of ethics and morality being involved into it. Now, what are different types of papers that are being considered into into the publication? So these are different types of paper which are being considered for publications: original research article, case report, okay, and then you also have something. Hello. Hello, am I audible? Hello, hello. Hello. Am I audible to you? Yes, yes. Okay. With the chat, I could. Okay. So, with the original research article is something the most important type of a paper, which provides with a novel new information based on original research and is supported by in-depth statistical analysis as well. The conclusions, what have been there, are supported by data provided into the results. We also have case reports, like we have PharmD students who have been publishing the case reports from different institutions, whereby description of a single case study with a unique features, and that may consist of something which is previously unreported observations, clinical conditions or results or methods or complications of a procedures or adverse reactions to a drug, which generally happens to be in case of a case report which is being published. Case reports are generally short and very focused one, and is often prescribed limit to the number of the figures and references in case of a case report. Review article, a detailed analysis of the recent development on a specific topic, maybe the vaccines to be delivered via nasal route, maybe the treatment, advanced treatment options for glaucoma. Okay, so on a specific topic, you will highlight the important points that have been previously reported into the literature. This type of a paper, like review article, would not introduce new information and even does not include author's opinion or a personal experiences. Okay, a large number of relevant references we expect into a review article. And reviews are many times even invited by the editors and also known as something you call it as invited reviews. And then you have something you call it as a letters to the editors. So an experts like Vanna madam or maybe Moria sir who have been into the field for years and have shown their expertise. Okay, so many journals have a letter to editor or correspondence sections which are generally short and can be written on any subject of interest to the reader, including a research study or a comments on a previously published articles. And these comments are highly objective and highly constructive, being written by the experts. Okay. Commentary is also something which is similar to the letter to the editor and commentary being there. Okay. Now coming to our part, what we call it as the original research article. The basic structure of a scientific paper is summarized by an acronym which is very well known by all of us is IMRAD, IMRAD, which stands for introduction. What question was asked, what problem was addressed being there into the introduction section of the paper. Materials and methods, so how the experimental studies were being carried out, how it was being studied. Results, so what did you obtain from the experimental studies, what was found by you and your co-authors and your team members being reported into results. And finally, how does this finding correlate with the current research, or what do the findings mean to all of us. So this basic IMRAD structure, IMRAD structure, is common to most of the standard scientific paper, specifically to the original research article, although there may be a minor variations depending upon the individual journals. But as of my experiences, most of the journal follows this basic structure of IMRAD. Okay, so introduction, methods, results, and discussion. And then what are other components of paper that include title page, abstract, keywords, tables, graphs, figure, conclusion, acknowledgement, and references. So we will start with one by one all different components and what are different tips to write down these different components, and how can you acquire, or how can you acquaint or adapt yourself in writing those different components of the paper. So with the first one, the title. It is the signpost which will tell our reader what your paper is about. It will encourage all our reader, all our scientists, what it is about, and it will also encourage them to invest their time to read your paper. So the title and abstract should be as compelling as possible. It should be as accurate as possible, because this is something which is going to give you the citation. This is something which is going to give you the wider outreach. Okay, the title and the abstract are in open access from all of the journal in most of the searchable database. So this should be as concise and as precise, informative, descriptive. It should not be misleading and unrepresentative, very specific and unambiguous. It should be interesting. As I said, it should be as compelling as possible, not too long, not too short as well. Okay, there are some limits on the words even in the title by the publisher, so you can obviously refer to the author's guideline. It should give an idea about the content of the paper, uh, and grammatically, obviously, it has to be correct with the syntax properly placed, and no jargon and no abbreviations are being expected into the title. Now, this is one of my recent article being published of my last to last year MPharm student, whereby we tried to load the acetazolamide drug with nanoparticle of acetazolamide into the contact lenses. And we have optimized a formulation using an experimental design. And the objective was to have a controlled ocular drug delivery. So as you can see, the title being put forward over here by our team is very specific, very concise also, with the name of the drug, with the name of the delivery system, with the name of the experimental design that is being there, and a very brief objectivity being there of controlled ocular drug delivery. Okay, so the main elements that what I feel should be included into a successful title is that we can try to have, I'll just give you all the references from where I have prepared my presentations as well. We will cite the main factors being studied. For example, drug. So the name of the drug acetazolamide in case of the title what I had shown you. The population or the clinical context being studied. Maybe you are studying a glaucoma as a disease or an acute myocardial infarction population or something like this. We have written the experimental design over there, whether it would be in your case a pharmacology epidemiological study, whether it would be a cohort study, randomized double-blind control study. Cite the main findings. So main finding is controlled ocular drug delivery was possible, or it is something increasing, manifesting, reducing can be put over there. We can put the most important aspect first. If the focus is on surgical intervention, if the focus is on chemotherapy intervention, so let us be there and make it as a distinguishing characteristic of your article from the other. And then you can start the title with the intervention name. We should always avoid imprecise formulation that serve no specific purpose. For example, a report of or the effect of, if there are effect of something like this. Use international common denomination of drug names. Commercial names should be avoided. For example, Calpol, Dolo, or something like this. Okay, they indicate the proprietary name of a pharmaceutical company and maybe constituted as an indirect bias or a preference to a particular company. So these brand name are not necessarily same across the different countries, and you know very well that your research paper will be read across the world. Subtitles should be used very rarely or sparingly. Reserve subtitles for names of study group, and specific recommendations may apply depending upon your target journal. Okay, so these are some take-home message of the important component of the title. Okay, coming to the very important point, next one is the abstract. The only part of the paper which is freely available through the search engine via electronic search engine, and it is read by many more people than the rest of the entire paper. Very crucial, very significant to sum up our paper in 150 to 2 words, which is generally a requirement from most of the publishers. Abstract is something, the model of the entire article or the model of the entire report, and it should describe all main elements of the research as per my experience, and it should encapsulate, or it should cover the research question and the research answer. And the abstract, what we find nowadays would generally be into two different ways being expected from us: structured and unstructured abstract, and even graphical abstract is also being asked by many of the publishers. So abstract should cover what was the research question, how did you investigate the question, and what did you find. So these all 3 points are there in your abstract. You make a checklist for yourself whenever you sit down and try to prepare your abstract. Okay, what was the research question? Okay, how did you investigate the question? So what was the research question is something, the statement of problem, or what problem you are addressing, or what would be the objectivity being there, and that by what you want to achieve. What were the experimental brief studies, and what were your important findings? So all these 3 points should be the basis of your paper. Good abstract would always describe the research objectivities and the finding. It is most of the time written into the past tense, covers only material discussed into the paper, contains no abbreviations and acronyms, and also includes keywords. As I was discussing, later on I would show you the structured abstract as well. Now this is the abstract of the title of the article. What I had shown you has been published into a paper recently in 2024. So whereby you can see the aim and objective being written, the material and method portions have been written, the major findings have been written, and the major conclusions that we have achieved have been also reported over here, with some of the finding quantitative absolutely have been written into this particular abstract. So by reading the abstract, the reader will be encouraged to read the in-depth full text citation or full text reading of your article to get some insights from the methods what you have reported in your research article, from the results that you have obtained in your research article, and which will give you the wider and a broader and a higher outreach to your scientific article. Structured and unstructured. So the previous one was very much unstructured. Otherwise, you may have objectivity, design, result, and discussions being there, as you can see over here. But this is not something a big game. like you have unstructured and you can convert into a structured form. You have written something, you start with a structured abstract so that you would be more clear, and then you can combine it together into a kind of a paragraph. Okay, so aim, objective, material, method, result, and conclusion. So that also all has to be very much concise and very much brief so that we complete it into 250 words as a good abstract. Now, something take home message, main points to keep in mind when you are writing the abstract. Okay. Background, that is what we can call it as a brief reminder to the context of the study, or brief statement of the main objective. Should be very, very short, up to 1 or 2 point only, into a 2 or 3 sentence. If possible, try to find out the gap in the knowledge. There can be a gap in the methodology, research gap into the results. Okay, there could be a research gap into the problem, how it has been addressed. Methods should be outlined with main inclusion criteria to define your sample population or something when you are dealing with an epidemiology or a certain pharmacology or a clinical papers. You won't have a time, or you won't have a space to explain all the methods in Greek in detail. So let us stick to the overall defining criteria. List the main results with mean odd ratio. Like as you have seen on my abstract, there were mean results with standard deviation and something being there. Conclusion should be there. Only a one-line conclusion summarizing your main finding is something which is sufficient. Please remember, no references into the abstract. There should be no discussion into the abstract, no judgmental statement into the abstract. Okay, something like surprisingly, contrary, we observe something. No figures or tables or other illustration into the abstract. That is sure that you may have the entire figure in form of a graphical abstract being there by many of the publishers. Next very important point comes is the keyword. This is something which would be there 3 to 10 keywords. It depends upon the author's guidelines to the publishers, how many keywords are being expected or under their guideline. These will capture your topics of the article. These are some of the words or which will assist the indexers, or which will assist the search optimization, search engine optimization, which is a terminology nowadays, a buzzword, SEO, search engine optimization. So this is something which will be helpful in cross-indexing of the article and are often published with the abstract. So a proper choice of keyword will help the paper to locate easily during a literature search, particularly an online search. So if your article is properly searched, you are going to get a wider outreach and a higher citation of your published article. In the previous title or the paper that I have been discussing along with it, these were some of the keywords what we tried to put up. Contact lens, Eudragid nanoparticles, name of the drug, name of the delivery system with the polymer name, okay, experimental design, and the name of the central composite design. So these were some of the keywords which we put into the keywords being there. Okay, so we have discussed keywords, we have discussed about the abstract, we have also discussed about the title. Now coming to the next segment of the and what are the tips which will be taken care of while writing down the introduction section or the introduction part of the research article, original article manuscript. Now, what is introduction? Is something which will give you the background or the context of the study. It reviews you on the knowledge of the topic. It justifies the need for the study, provide information on the study. So organization of content will start from the broad issues. I'll just give you with the example of my published another article, how broader issues are being narrowed down to the immediate problem, and what is the need and justification for the study, what we are reporting in this paper, and what are the objective or the hypothesis of study. So please remember, the objective of your research work will be there in the introduction section of your manuscript. Okay, like we may have an entire different chapter of aim, objective, rational, and statement of problem into a dissertation or a thesis, but we have something over here in the introduction section. So in introduction section, we have first is the study background, which introduces the study topic. It starts with a broader issues and will move down to the immediate problem. For example, antenatal care is essential ingredient in reducing the infant mortality. immediate problem, something you can narrow it down. Though India has national antenatal program since 1980, but no data are available to show the effect of this particular program. Okay, like we have a social and preventive pharmacy program in our B Farm, and whereby we are teaching the student different uh, health related programs or the government schemes. So this was something which I hope I could connect and correlate with your workplace as well. Relevant papers should be cited into the introduction section about the prior art, about the reviewing the knowledge or the review of literature. For example, ABC XYZ 1 to 4. So 1 to 4 represent the citations into the text. Please remember, do cite only papers which are relevant to your specific research topic. Do not cite well-known facts. For example, maternal mortality rate is higher in developing countries than in Europe. Okay, so we'll just talk about reviewing the knowledge in the example. I'll show you how introduction have been written by us for our one of the published paper. The justification will follow the general and immediate problem and review of the current knowledge that supports the need for the study. For example, during our literature search, toxicity data of drug X was not available. Hence, we planned to conduct a toxicity study and something like this. As there is no data of life of a diabetic patients, we planned or we ensured to have something like this. Okay, how are you justifying the need of your study? Okay, your was about investigation on quality of life of diabetic patient. Your study was to find out the toxicity study data of drug X, and this is how we are trying to justify the need of our study. General type of study, such as what you call it as will be there in introduction, general type and objectivity of the study. Like we conducted an experiment. Okay, we surveyed the attitude and the practices. Okay, we conducted an experiment to see whether the animal fed drug in a controlled environment. So this is how you can plan to write down the introduction. Now, what not to include, please remember, this is something which is very, very important. I'll not just try to highlight you what to do, but also try to highlight you what not to do when you're writing a research manuscript. So please do not include any information that is not directly relevant to your study. Please do not include any information that is not directly relevant to your study. We have a limited number of pages or the length prescribed for the introduction section. This is not going to be the history. This is not going to be the history page or a story page of your particular article. So problems other than the one being examined, please do not include. Any general information about the country, any general information about the hospital, any general information about nanoparticle preparations and evaluation and characterization, no need to include. Details of the research design also we are not supposed to include into the introduction section. This is something which all of us should take care, because see, once you will start writing it down, you are very new on writing it down, and you may be disheartened once it is rejected from the editor side only before it goes into the peer review process. Okay, so we need to take very much care. An introduction, obviously, at the to summarize this particular section is something, it should answer the following thing: problem, previous efforts dealt with it, as well as what remains to be done, what are objective and hypothesis of your work should be there in your introduction section. Now, what are different aspects that you need to take care whenever you are writing an introduction section? The take-home message: background describing what is known on the subject. For example, percutaneous coronary intervention is the cornerstone of therapy of acute coronary syndrome, but may be associated with the procedure-related complication. What is not known? What elements are still to be investigated, or what under different methodologies are under controversies? What is the exact research gap in the knowledge that your study will try to fill up those gaps? Can we cite certain existing data, especially conflicting data that indicate uncertainty? So as I have shown you on some previous slide, it remains unknown whether to date it has not been proven no study to date has been investigated the effect of drug X on Y. There are few data to quantify. The effect of X on Y remains unclear with the published literature. Objectivity would be there, plus minus the working hypothesis. Can we hypothesize that the administration of therapeutic lens would reduce or would increase the ocular bioavailability in context of better therapeutic outcome for glaucoma treatment? Cite the exact parameter you plan to measure. We aimed to improve the ocular bioavailability. We aim to assess the or we aim to investigate process parameters on the nanoparticle entrapment or nanoparticle certain more parameter. Cite the type of the patient population or clinical context, those who have been there on the pharmacology or a clinical side, and cite any secondary objective if it is being there. So this is something, a take home message to write down the introduction section. Okay. Now, many times when as previously also I have delivered a lecture on this particular topic where the query was what should be the tense. So with the time limit, see, to describe the current state of knowledge, always you write down into the present tense. Cancer is a common disease. To describe the observations previously published by the others, past imperfect. So Smith and his co-workers show that the drug A reduced the rate of the death where drug B did not. To describe a process that began at some unspecified time in the past and is not yet complete. So you can always do it into the present perfect. So like several researchers have investigated the effect of drug A onto the disease. To describe something that has not happened yet, so present perfect is the right tense to write down it, something like it has not yet been determined whether. To formulate a hypothesis, past tense for first verb and present tense for second verb. Very smartly, you can put it up like we hypothesized. So past tense for the first verb, that drug A increases, present tense for the second verb. So this is how something you can write down the tense. We do not have any specific rule of thumb that entire introduction section should be written in past tense or in tense or present perfect tense or something like this. To formulate your objective, past tense, we aim to measure something like this being there. Now, this is something, an article which has been published. He was our MPharm student, Anjum. Along with me, Dr. Furkan and me both have guided him. And this is the title of the article. This was the abstract. I have already shown you the abstract of the previous article. Okay. Now, coming to the introduction section, how we have written in our published literature, what I had shown you on this previous slide, this is the article. You can see we started with a broader issues with the problem. These are the text what you will find into my published article. Most ophthalmic drugs are delivered through eye drops. What are major limitations of eye drops? So we started with a broader issues of the problem that we are addressing through our research work. Now, what is the solution to the problem cited being reported into the literature? I am repeating. What are how the problem was addressed through the prior art, through the literature that has been published. So NDDS has been advanced to improve the residence time of the drug and penetration through the cornea, but they have their own limitation. So obviously, I will justify my need that a need for NDDS that can increase the drug bioavailability, minimize the systemic exposure, and improve the patient adherence for better clinical outcome. So I justified that drug delivery through contact lenses is one of the alternative, and there are advantages of contact lenses being reported in our article. Then we have shown in what is been reported by previous scientists or published work, sustained release of dexamethasone from silicone hydrogel contact lenses. There is someone who has also explored the molecular imprinting technology. Okay. Kapoor and his co-workers have also tried something. Limitations on the method of the lens preparation is faster release due to diffusion of drug from the contact lenses. There are authors who have also prepared particle-laden P hemosheets. There are authors who have incorporated methacrylated hyaluronic acid into the contact lenses, improve the hydrophilicity. Okay, so these were some of the solutions to the problem cited, which are there in your which is there in your work. Now, what are the disadvantages of these solutions being addressed? Okay, so whereby we have smartly said that the work is there, but it significantly enhanced the cell interaction, but it decreased protein absorption and thus improved biocompatibility and other material. Comfort of contact lens by incorporating HA as a wetting agent. Researchers have controlled therapeutic lens using nanoparticle, micelle, microemulsion, use of vitamin E. These technologies showed sustained drug delivery but affected the physical and optical properties of contact lens. So over here you can see on my last point whereby we have tried to address the limitations to the problem that have been cited. I hope I'm making a sense over here and I'm able to connect with you all with the first section, that is introduction being written. Now something more about the diseased literature being there. You can just put up the questions over here. I'll be happy to take down the questions at the end of the session also. The statement of problem: more than 50 million people are affected. Symptom of dry eye significantly affect the quality of the patient life. Treatment of dry eye being reported about the disease literature. The limitations of the eye drop and something being there. So then finally we come down on winding up our introduction section of the research manuscript. Okay, and I'm going bit of fast, but as I have to cover much of it, so just you let me know by unmuting yourself. I'll be just be trying to be a very brief, very slow, or I will pace down a little bit. Okay, thank you. So as we said, introduction part also covers the objectives and the hypothesis. So we have written in our research paper that we are here to improve the patient adherence and better clinical outcome by developing an extended release hyaluronic acid implant hydrogel lenses that will be designed or developed using a 3:2 full factorial design to optimize our formulation, whereby we will try to achieve minimum leaching of the drug during sterilization, as it has to be administered into the ocular cavity. It has to be a sterile. Maximum sustained release, we will try to tailor the content of one of our excipient EGDMA and the thick. We have written the final aim to develop a HA-ridden implant hydrogel contact lens for sustained ophthalmic drug delivery without compromising optical and physical properties of the contact lens. So I am supposed to produce an empirical data, empirical evidence whereby optical and physical property of contact lenses are not compromised. I am supposed to give the data where HA-laden ring implant hydrogel contact lenses have been designed and characterized. I am supposed to provide empirical evidence, result and discussion portion whereby the drug release have been into the sustained fashion. So your introduction is having objective and hypothesis that has to be connected or correlated with the result and discussion session. Okay. Next particular part being there is about the method section. Excuse me. This section will answer who, what, why, when, and where of the study. So what is the purpose of the method section? This is going to describe how the study has been designed, how it has been conducted, and it will allow the reader to judge the validity of the procedure. Now, this is something, a very important part of your paper, which is going to give you the citation. Okay, because there are many people who are going to cite you that I have conducted the muco-adhesive strength study as per so and so author. I have conducted the in vitro drug release study via using dialysis bag or membrane using so and so cutoff membrane size as particular author. Okay, so it will allow reader to judge the validity of the procedure also, and it will give you the outreach of the article as well. It will provide information also on the replicate study to you. Okay, so this is something which is very, very important, whereby all of us should focus much on writing down this particular section. How the study was carried out. So we are supposed to provide accurate details of the materials and the procedures being used, details about interventions, how the dose was administered, what was the route of administration, study implementation, exact drug dosage, exact form of the treatment, exact details of unusual apparatus being there. Okay, maybe you have designed your, like we have our own procedure we developed to conduct the ion permeability study via conductivity study of sodium chloride. Some of you might have also seen an in-house developed mucoadhesive strength determination apparatus. So you might also have your in-house modified diffusion cell apparatus. So we are supposed to provide details of all those unusual apparatus being there. Apart from it, you are also supposed to provide the statistical test being used over here, computer software, the name of the software and the details. Let it be SPSS, let it be Design Expert, let it be other software which is being used in pharmacokinetic data analysis. What are the planned comparisons between the groups? Are we showing the significance level to disapprove the null hypothesis? Yes. We are supposed to use very, very short and simple sentences, and we will be very precise. Acronyms and abbreviations can be used in this particular section of the manuscript, and we are not supposed to include the background informations and the result over here, because this is purely your area, what you have done. Okay. A good method section should answer the following thing. Whether the text what you have written in your experimental part, what we call it as a material and method, is describing the objective what is there in your introduction, is describing the question that is being asked, what was being tested, how trustworthy the measurements are being there under the consideration would be. Okay, have you carried out the measurement by simply performing certain titrimetry method, or by UV spectrophotometry method, or how did you perform the adhesiveness strength of your gel? So that all is answering being there or not. Were these measurement recorded, analyzed and interpreted correctly or not? Would a suitable qualified reader be able to repeat the experiment in same way? This is something I hope personally the need of the hour, because like on day one, many of you and including I would also agree with you that we find that the papers are not reproducible. So can we write a material and method section in such a fashion that our colleagues, our peers can repeat the experiment into the same way? So this is something which is very, very important. Now, the basic tips on is that you start writing early during the study before you begin the study, so that you will be even very clear when you are performing the experiment. So write it down in your laboratory notebook, in your journal, in your record book, that I am going to perform in vitro drug release study by so and so method. So would be the analytical method for study for analyzing the drug in my dissolution sample. I will also at the same take care that I will avoid micro detailing of all the procedures. The usual order of presentation should always be chronological. Okay. However, related methods may need to be described altogether differently. I hope you understand by meaning over here that how they are supposed to be written in a chronological order. Like you start with pre-formulation kind of a thing, or certain preliminary batches, preparations, and then you write down characterization method. So that in that particular sense, I'm conveying you that we are supposed to put it into a chronological order. We are supposed to describe the measurement precisely and include the error of the measurements being there. We are supposed to use preferably active voice rather than passive voice whenever and wherever possible. Okay, write what we have performed, what we have conducted, and not something what we intended to do but we could not do. So let us be honest and be ethical, that we should write only what we have done and what we have conducted and what we have performed. Cell line study हमें करना चाहते थे, हो नहीं पाया तो मत लिखिए उसके बारे में. So whatever you have done only, that you only write and report over there. Next is some more tips associated with it is describe measurement precisely and include the error of measurement. Write with precision, clarity and economy. For example, every sentence should convey the exact truth as simply as possible. Show the section to all your co-authors, or MPharm student, PhD student, your colleagues. ask if they would have difficulty in repeating the study, then you are supposed to rewrite your material and method section. This is a very simple litmus test for all of us. What you have written, you ask your colleague to read it and let him explain what he has understood from it. Can he follow the procedure what you have written? If he says yes, you can surely go ahead with the first draft of it. If he finds difficulty in repeating the study, you are supposed to reframe and rewrite your experimental sections or the method sections being there. Now, this is something what I have presented it, list of items to be included specifically for those who are working in clinical or associated with PharmD students and pharmacology and clinical pharmacy. So retrospective studies, something which we are supposed to write down into material and method section, inclusion and non-inclusion criteria, ethical consideration, primary endpoint, secondary endpoint, okay, statistical analysis, data recorded, source of study data, subgroup, number of subjects and samples. For prospective study registration, if randomized, what is the sample size calculation? This is something which is very, very important. Randomization procedures also and intervention. So this is something which is very, very important take home message for the material and method. Continuing with the same article, I'm just trying to highlight you. See now, this is something what you find over here is that hyaluronic molecular weight has been written. IUPAC name of all chemicals have been written. Okay, this would be a generic name or a trade name. This is an abbreviation, full form has been written. The same way over here. Okay, ultra pure water of a certain conductivity have been written over here. The sources of material have been written over here. Okay, so you are supposed to write very clearly about the materials that you have written. This is the very same part what has been written in this particular article which has been published by us. Okay, material and method will be there as composition of pre-monomer mixture for hydrogel contact lenses and implant. This is something how we have tried to show it into our article. You can see methods about formulation and development. The full factorial design matrix with their variables and their labels have been there with independent variable, dependent variable, along with their limit. We have tried to show it into our article. Okay, and the regression equation as well. So preliminary experiments, the concentration of AGDMA below resulted in an implant. Like I could read one of the question that should we add the preliminary studies performed for the selection of variables. So this is how I have shown it into our article. We have shown it concentration of below 0.5 resulted in an implant with poor mechanical property. The concentration greater than 1.5 resulted in a brittle and hard implant. And that was the reason for selecting the range for this particular excipient or the label for that particular independent variable. This is being ranged between 40 to 80 micrometer to give a total thickness of contact lens between 80 to 120 micrometer. So very briefly, like one of the participants have asked that how do we try to connect our preliminary experiments with the main experiment. Now, this is something, a study which has been performed, okay, where contact lens is prepared by soaking method or immersion method, what you can see, okay. You can see over here the past tense most of the time have been used over here because the study has been conducted and have been completed. Okay, all the batches were prepared along with the checkpoint batch analysis. Okay, so I'll just go through it very fast. Now this is a figure and illustration. I'll come down to that particular part also at the end for structuring your manuscript. Schematic diagram of fabrication of hyaluronic acid leaden ring implant hydrogel contact lenses. So how illustrations has been done and have been prepared using different tools available online. The TS of the implanted ring in hydrogel contact lens has been taken and the photograph have been mentioned over here. Okay, a very raw crude data have been also being mentioned over here. The entire procedures for all different experimental study. How did we perform the sterilization study and sterility testing? How did we perform the ion permeability or ion diffusivity study? Okay, you can see how did we perform the in vitro release kinetic study. The entire procedures have been written. The simulated tear fluid was replaced at every interval with 2 ml of fresh simulated tear fluid to maintain the perfect sink condition. So this is how we have tried to break up the each individual study in a chronological order as I showed you, as I described you slide. without affecting the critical properties of the lens. So effect of packaging solution, it was stored in so packaging solutions, and at the interval of 3 and 6 months, they were removed and analyzed for the leach out of the drug. How did we analyze the drug? How did we estimate the dose to be incorporated into the lens? So along with the reference we have cited into it. Okay. Entirely one study has been described separately. The reason for taking this particular paper as a case study for the today's session is that we do have kinetic study, we do have dynamic study, we do have in vitro in vivo correlation into this particular paper altogether. So what was the purpose of conducting the kinetic study? What was the procedure? The approval of IAAC has been also being written. So I have just tried to put it into the bullet point from the text paragraph point written into our article being published. So you can see these were the quantitative parameters that were being resulted into Cmax, Tmax, and mean residence time. In vivo efficacy study, dry eye syndrome was induced. It was induced with which particular agent? Benzalkonium chloride. Corneal damages were being scored with absent and something. So we have over here reference also for the scale which is being used. How would be reporting it to a result if the score is greater than 3 are considered as a dry eye induced. So I hope you will agree that we are finding it, the manuscript which is being written is something which is as clear, as concise, and as precise possible. Images were taken at different time interval till the 10th day in order to rank our stain level. 2 microliter of 1% fluorescein sodium was instilled into the set. Okay. Entire statistical analysis, like when I said material method, we are supposed to give you the details of statistical analysis. It was carried out using SPSS version 21. 0.0 for Windows, and t-test two-tailed and one-way analysis ANOVA was used to compare different group after confirming the normality and homogeneity of the variance. These are assumptions of these two hypothesis testing of ANOVA as well as a t-test. So we are supposed to write down all the details. I hope I'm making a sense on writing down the material and method section. Before we end the session, I'll just give you the recap and the flash back of the entire writing an effective research paper with structuring of your manuscript. The purpose of result section, we are here to demonstrate the integrity of the experimental studies that we have undertaken in our research paper. We are supposed to provide the outline to the answer to the research questions, what you have written in your introduction section by presenting the results of analysis, as I was highlighting the introduction section of this particular paper. We are supposed to provide and present the data on extent to which the study was actually implemented and planned. For example, in a clinical trial, all patients were supposed to receive the treatment, but about 96% received the experimental or intervention and 93% of the control group received the treatment. So you are supposed to give the actual study about it. Maybe you started with 6 animals to be administered with a lens to be inserted, rabbit, and all of a sudden 2 rabbit have fallen out of the study because they were ejecting the lens out of the eye. We are supposed to put it down in some way that only 4 animals have been studied over here, or the data have been shown out of it. Present the main results in a pre-specified objectives, hypothesis, or a planned comparisons. Present the data, present only those data that demonstrate the results of relevant experiment. तो यहाँ पे FTIR, melting point जैसे थीसिस में होता है, वो सब डेटा लगाने का जरूरत नहीं है. जो आपके ऑब्जेक्टिव में, like you have seen in my thing, ion diffusivity, in vitro release kinetic, or maybe the kinetic study and dynamic study. So those relevant experiment results are to be there. Do not present the information only because it was just collected. It has to be connected with addressing the problem, with addressing the objectivity, what you have written. Commenting on the data in the results section should be restricted. We should limit the comment to the statement about magnitude. We should only write about the magnitude or the direction of the differences or to qualify the finding. We are not here to editorialize our own content. Commenting on the data in result section with interpreting the result would be there in discussion section, so not in the result section. Many of the articles would have result and discussions, publishers would have result and discussions together going on. Most of the journal would have separate result and would have separate discussion portions being there. Now, how do you present the results? Either you present it into a textual form in a paragraph way, you present it into a tabular form, you present it into a figure or a graphic form. Okay, I'll just take you very fast.
Audience, just wait. There is some internet issue, I guess. Just wait. I think Cyrus get disconnected due to some network issue. Just reconnect with him and get back to you soon. Just wait.
Hello, am I audible now?
Yes, sir. Yes, they're audible
Am
now.
I audible?
Yes, sir.
Sorry,
Yes, sir.
we lost yeah, fine. So I'll just continue with where I have left. My visible?
Yes, sir.
Yes, sir. Can anyone tell me what is the title of the slide?
It's figures and graphs.
Figures and graphs. Okay, fine, fine. So I'll just continue with where we have left. Okay. So the sum checklist for the table, it should be short, very self-explanatory caption. Units of the data are given there on the table. Please verify number of subjects, elements, how many times you have replicated your study or dissolution. So n is written or there or not. Mean plus minus standard deviation, mean plus minus SEM, okay, are given in there or not. Statistical significance of group are indicated by asterisk or some by other means is not there. P values have been given in there or not. The results of in vitro release kinetic are as reported in table 4. So have you cited into the text or not is also very, very important. You put up a table, but there is no mention of the name of the table or number of the table in the manuscript. Not a repetition of the text we need to please take into consideration. Otherwise, it is unnecessarily going to increase the length of your manuscript. The table format obviously has to be as per the target journal format. Okay. Figure and graph. We generally show you the figure and the graph into our manuscript to show the trend, to show the amount, to show the frequencies, and to show the association between the data. And where we are supposed to prepare it into the format as required by the journal. Okay. So as you can see over here, effect of drug S on diastolic blood pressure in different age group. All different age group have been with the different color coding. You can see the plasma concentration time curve of reference and a test formulation of cyclosporine being reported in one of the published article. I'll just come back to again this particular portion before winding up the session. Again, the checklist for the figures which are there in your results. Self-explanatory captions should be given and should be mentioned again over here. If it is X and Y axis are graduated, titled legend are there, units are mentioned, different symbols and markers for different group. As you can see, we had a different symbols for different age group. SD or SEM represented graphically or not, vertical bars over here. Okay, statistical significance indicated, data given in table or text repeated. If it is repeated, you are supposed to eliminate either table or a figure. You need to take a call upon that. I'll just discuss when should we consider that. Now, next particular portion after discussing the result section is something very important is the discussion. Now, this is your opportunity. This is your science and art and expertise to convince the reader regarding the importance of the finding. Okay. So I'll just, okay. Importance of findings and to influence further research and applications of our finding what we have taken. This particular part of our paper is something more than a summary of the result section and more than a list of recommendation. Please remember these 2 points. It is something more than the summary of the result section and more than the list of a recommendation. Okay. So let us be honest. Let us very straightforward put the limitations of our study. Let us also discuss the implications of our finding and also explain the reader, the scientific community, why our findings are very, very different from the findings what are reported into the literature. Okay, so what we should not do is that we should not introduce new data or analysis which is not there into the result section. Please do not make claims or over claims which are not supported by your data. I am repeating, do not make any claim which is not supported uh, by our data. You can discuss some of the part people argue that we can discuss some of the result experimental work which is in progress, but I personally advise that we should never ever discuss whatever other research work or the research finding that is into the progress. For example, the student have passed out, but still we are doing some of its kinetic study, dynamic study, cell line study, or some more in-depth study on characterization, physical characterization with interfacial tension, or maybe a dynamic surface tension or something else. So we are not supposed to highlight it into this particular part. Okay. Can we organize and format the discussion section? For example, like we may have the main conclusion section being there. Okay. Brief summary of most important evidence, limitations of the study. Many of the participants are asking for the PPT. I would surely pass on to the organizer, but this PPT would be you would be able to connect it if you would listen right now and understand it and comprehend something out of it, because there are many things which are not written onto the PPT, and I'm trying to share something based on my experiences while publishing or while guiding my students. Brief summary of most important evidence, limitations of the study, findings which are consistent or inconsistent with the results reported earlier, implications for theory, policy, and program, and conclusion. So you yourself start dividing it into the different subheadings of the discussion, and then you can combine it into a paragraph form. So this is something which is very, very important. This is something what I have been saying, more than a summary and more than a list of recommendation when you write down the discussion. So how we have something as a main conclusion. Okay, so first statement of your discussion can be started with main conclusion. This study conclude that. Okay, second can summarize the data supporting the main conclusion. Approximately 38% of subjects were found to have so and so, and nearly 22% had no more than something like this. We could not do something due to lack of facility in our institution. We also did not do, and thus are not sure. Like recently, we have clearly written that we could not do the SAM and TAM study for our micro emulsion leaden contact lens, which is right now under the communication with one of the publishers. Okay, so you can surely write down the limitations. You can also write down the comparisons with other research are finding consistent or inconsistent. Unlike other studies, we found this finding may be due to the fact that other studies did not highlight the implications. Like the study suggests that a single dose of drug X should be added to the standard drug Y for the treatment of, and something like this. You can write down the implications, comparisons, limitations, and conclusion. So how have we written the optimization of the same paper? I'll continue with the result and discussion section. Okay, so the result, like you can see what we have written over here. Okay, these are the data what we have shown. Okay, the cross-linking of acrylate polymer metric increases with increase in the level of EGDMA. and which result in entanglement of HA polymer in implant matrix. So this is something which has been discussed about the trend that we observed into our result part. With increase in thickness of the ring, the percentage leaching of HA from implant contact decreased. Why? Because of increase in path length of the drug to permeate through the hydrogel matrix. The interaction effects were not particularly being significant. We may have some another day a session on interpretation and analysis of the experimental design. Okay, so I'll just keeping it right now in the best interest of the time what we have. The results also suggest the thickness of implant has predominant effect on retention of hyaluronic acid in comparison to the cross-linker. So out of the two variable, which is the variable which is mainly responsible for it. So this is how we have shown the data analysis of sodium ion permeability and diffusivity at the time required to release 50% drug was significantly affected by the thickness of implant in comparison to the EGDMA. You can see how we have written n is equal to 6 mean plus minus standard deviation, time in hours, percentage drug release on y-axis, and with the bars also being reported of the standard deviation. So this is a completely a reported data into the article which has been published. A line has been there for a therapeutic outcome. So above this red line, everything is comes under the therapeutic dose. Below this is something which is a subtherapeutic. So the drug release nanogram of the drug release per hour from hyaluronic acid from implant contact using cumulative release data. We have also described are shown as a mean plus standard deviation. We have conducted the study replicated 6 times. Therapeutic level indicates minimum therapeutic drug release rate. So this is something 83.3 nanogram per hour required, considering 1% absorption from 0.1% weight by volume hyaluronic acid eye drop. Now this is something very, very important. My control batch is eye drop and my formulated drug device is the drug delivery system which is there being laden into the which is nanoparticle, and then it has been laden into the contact lens. So this is how we have tried to show. The burst release has been discussed, the sustained release has been discussed, the mechanism of the drug transport and entanglement into the chain matrix has been discussed. Okay, so the diffusion coefficient of the contact lens has been there with suggesting the control release. Anomalous behavior, I hope all of you know the model dependent release kinetic, a low value of K, significant enhancement in release duration with HA leaden ring implant lenses into the comparison being there. Okay, so we have tried to discuss the results with the trend being observed with the cause and the reasons being associated with it. Now, this is the profile of the drug release which has been stored into the packaging solution for 3 and 6 months. You can see the ligand being there. What is there at the initial stage, what is there at the 3 month, it is exactly overlapping. So there was no leaching, almost equal leaching even compared to the time being there. Okay, so showed an initial burst release and lens diversion average concentration. So AUC has been also repeated into the textual form. Now this particular part has been magnified and have been reported as a sub graph over here. So this is also quite interesting. Within the graph, we have also included the experimental animal and the delivery system photograph into our published paper. So this is something what you can notice it into when you read the articles which are being published. Okay, this is a Levy plot for in vitro in vivo correlation between the in vivo cumulative percentage of drug in tear fluid and percentage of drug released in vitro with a high correlation of 0.9961 and 0.9774 for both of the dosage form that we have taken. So this is something what we write about the discussion related to the in vivo efficacy study. Healing into the rabbit eye treated with HA implant contact lens is faster in comparison to the positive control group, which worsened over a period of a time. Staining degrees were significant. You can see over here the p values being shown. The corneal epithelial defect were observed during the initial days, but were due to benzalkonium chloride. The right eye showed progressive decrease. So untreated left eye, no healing was observed during the entire study period. Okay, so you see the time period as well as the efficacy study regarding the dry eye study induced the dry eye using benzalkonium chloride, and over a period of days how the conditions have been worsened or have been treated being shown over here. Okay, and then finally we wrote our conclusion. In many of the journals, conclusion cannot be a separate part, but then conclusion can be a last paragraph in your discussion section as well. Okay, so it's almost 30. We have around 30 to 40 more minutes, whereby we will try to now discuss something where uh, will try to help you how you can organize your manuscript and the work. We have seen different sections over here. I hope, am I audible? Samresh sir?
Yes, sir, loud and clear.
Hello? Yeah, loud and clear. Okay, fine. Now, I would like to just discuss about a stepwise procedure for structuring a research work, what they have at their workplace, and whereby these work something where editorials can be taken seriously. Okay. Now, this is based upon my experience. This is based upon the reported methodology also, that how should we start converting our work into a manuscript. This is something which is very, very important. And this is something a recap of what we have studied already. Okay. Or what we have discussed already. Okay. Fine. All of you are there with me. Ready? So first thing is that you prepare the figures and the table. Okay, so what do we mean by preparing the figures and the table is that see, before you start on preparing your manuscript, I'll tell you that the topic to what you have studied or the problem what you have studied has been resolved and solved, or you are on the verge of completion of the solution, and you should define your hypothesis and objective. Okay, because as they are going to be there into the introduction section, which is the step 6. But also I would request that you review the literature related to the topic as well as some of the more selected papers, around 25 to 30 papers that can be cited in your paper, and this will be listed into your step number 11. Okay, so what is the first step? First step is a preparing the figures and tables. Then next step you should do is write down the math experimental portion. Next step you write down the result. Next step you write down the discussion. Next step you write down the conclusion, and then you write down the introduction, abstract, and title, and keyword, acknowledgement, and reference. Okay. Right now, what we have discussed in previous 70 to 90 minutes is about the different sections associated with writing an effective research paper. But now, how can we organize our research work into a manuscript, and from where we should start? So first, this is something is figure and table. So figure is something 1,000 words. So any illustration, including tables and figures, are the most efficient way to present our result. And we should be very much in an impression that it is very critical and very crucial as well. How can we decide between presenting your data as tables or figures? Okay, then generally the table give you the actual experimental results, while figures are often used to compare of experimental result with those of previous work or with the calculated or theoretical values. So whatever is your choice, okay, no illustration should be duplicated, the information described elsewhere into the manuscript. Figure and table legend must be self-explanatory. Now what do you see over here is that we see over here an example that same data presented as table and same data presented as figure. So now depending upon your objective, we can show our data. Hello.
Yes, sir,
Hello, am I
audible.
audible? Yeah. We can show our data either as table if we wish to stress or emphasize the numbers, or we can show it into the figure if we wish to compare the ingredient. So ecological group station, okay, 1, 2, 3, 4, 5, and same way over here 1, 2, 3, 4, 5 can be there. So whatever your choice is, no illustration, please remember, should duplicate the information described elsewhere in our manuscript. Okay, so this is something which is very, very important. Now another very important is that figure and table obviously have to be self-explanatory. explanatory. Okay. Now, when you are presenting the figure and when you start plotting down the figure, please remember avoid crowded plots also, or of the so many thing or something which is being nicely being seen over here. You can see everything, something with asterisk black color, something with gray color water bodies with basic structure. Dotted line shows the basic coastal baseline, inner and external part. So this has been also taken from one of the published literature from the publisher. Okay. Now, we should avoid the crowded plots as you can see on this figure. Using only 3 or 4 data set per figure, use well selected scales also. Okay. Please give thinking about the appropriate axis label size. We are supposed to include whenever you are plotting the figure, include clear symbols and data sets that are easy to distinguish. Okay, so as you can see over here, we are cluttering so many charts with too much of data. So this is not something which is recommended or which is not being advisable. Please remember. Now, next very thing is that if we are going to use photographs and figures, I would advise to use color only when necessary when submitting to a print publication, because it makes you a heavy burden on charges of EPC. If different line styles can clarify the meaning, never use colors or other thrilling effect, or you will be charged with expensive fees. You can see this one and this one. Okay. This is quite possible with a black and white being there. Okay. So please remember this. But obviously, this does not apply to the online journals, because online journals, you can submit duplicate figures, one in color for the online version of the version and PDF, and another in black for the hard copy journal. Okay, so this also makes a common sense. Another very common problem is the misuse of line chart and histogram. Like we come across our MPharm student that they are not properly presenting the data. Okay, so the lines and the histogram, lines joining data only can be used when you are presenting time series or a consecutive sample data. But however, when there is no connection between samples or there is not a gradient, we must use the histogram. So please remember, otherwise, from editor point of view, or they will come to know that he is someone who has to work upon the figures and the graphs or the knowledge about it. Now, sometimes the fonts are too small for the journal. You must take into consideration they must be or they may be illegible to the readers. Please remember, figures are not eye charts. Okay, make them large enough to read. Okay, you must pay attention to use the decimal to lines and etc. So this is something on my part. I'm not touching upon over here what are different AI tools and online tools available to create the figures and illustrations and graphs or something. Okay. Next step after plotting down the figures and table. So what is table is very very important. Table आपने बना दिया तो समझ लो कि आपको पता चल गया कि कितना काम हुआ है और क्या हमारे पास एक्सपेरिमेंटल डेटा है. अब वो जो टेबल में जो डेटा लिखे हैं वो डेटा हमने कैसे प्रोड्यूस किए हैं, कौन सा एक्सपेरिमेंट करके, इन विट्रो रिलीज का एक्सपेरिमेंट करके, क्या हमने इंटरफेशियल टेंशन का एक्सपेरिमेंट किया है, क्या हमने एडेसिवनेस और म्यूको एडेसिव स्ट्रेंथ का एक्सपेरिमेंट किया है, क्या हमने स्वेलिंग देखा है अपना गैस्ट्रोरिटेंटिव टैबलेट का. सो समथिंग दैट. ओके, now what is the next thing is that now you start writing the methods. So do not repeat the details of all established methods. Like you are not supposed to write down again the entire procedure of drug release. You can simply write down that drug release was performed as per the pharmacopoeial method. Okay, so and you can give the reference. Okay, in vitro diffusion study was performed as per the published literature. Okay, use references and supporting material to indicate the previously published procedure. This is very very important. Broad summaries or key references are are sufficient. Okay, so this is something which you need to take into consideration. Reviewers will criticize incomplete or incorrect methods, incorrect descriptions, and they may recommend rejection. Please remember. So this section is very, very important because this section is critical in process of reproducing your investigation. आपका एक्सपेरिमेंटल मेथड जो है वहां पे ही लोग सीखेंगे कि मुझे नैनो पार्टिकल कैसे बनाना है, माइक्रो इमल्सन कैसे बनाना है, लाइपोसोम कौन से कंपोजिशन से बनाना है। अगर वो क्लियरली नहीं है तो वो रिव्यूअर उस एंड पे आपको रिजेक्ट कर सकता है। ओके, सो प्लीज यू हैव टू बी वेरी वेरी केयरफुल in writing down the entire thing. Okay, do not use proprietary and unidentifiable compounds being there written. Okay, for example, for chemicals you are supposed to write down IUPAC name, you are supposed to write down the generic name as per biochemical nomenclature. For species you have to write down the species name and number as per ATCC and different aspects. Otherwise you have to write down the cutoff size, you have to write down the molecular weight. ने यूज चीतोसन का यूज किया है तो कौन सा चीतोसन यूज किया है आपने, लो मोलिकुलर वेट, हाई मोलिकुलर वेट, you are supposed to write down the unit of measurement, and preferably I would advise you use the SI unit. Okay, so also present the control experiment and statistics. Like I shown you, eye drop was my control experiment. Okay, again make the experiment of investigation which is being repeatable. List the methods in the same order they will appear in the result section. आपने सबसे पहले स्वेलिंग स्टडी दिखाया है तो रिजल्ट डिस्कशन में भी पहला स्वेलिंग का रिजल्ट होना चाहिए. दिस इज द लॉजिकल ऑर्डर इन विच यू डिड द रिसर्च. ओके, नेक्स्ट इज something what we call step 3 is about not step 3 but writing down the methods only. Over here is that when we are writing the methods, please be careful. Now you can take a snapshot of this particular slide or a photograph, screenshot. This is very very important. What I would say, to look at the journal's guide for authors, but an ideal length for a manuscript is 25 to 40 pages, double spacing, including essential data only, not including the supplement data. And here is the general guideline what I have come across. Title short and informative, abstract one paragraph of less than 250 words, introduction should cover 1.5 to 2 pages, method should be of 3 pages, 2 to 3, result 6 to 8 pages, discussion 4 to 6, conclusion, figure, table and references. Okay, so around 50 references are there, so 4 pages. Okay, 4 plus 3 7 plus 8 15 16 22 30 33 35, okay, and 36. So around 35 to 40 pages you will wind up your manuscript. So this is something you please take into consideration. We have reached over here. ओके, मेथड्स हमने ये फिगर और टेबल का डिस्कस किया. नेक्स्ट स्टेप हमने एक्सपेरिमेंटल मेथड्स लिखा है, तो वो 2 से 3 पेज में सब हमें खत्म कर देना है. नेक्स्ट इज अबाउट राइटिंग द result. Okay, now this section is something which answers to the question what you have found. क्या आपने फाइंड किया है भाई. So only representative results from your research should be presented over here. The results should be essential, something which is for the discussion. Okay, but please remember that most journals offer the possibility of adding the supporting materials, so use them freely for data of secondary importance. Okay, like one of the reviewer asked us to provide us a calibration curve data, and we have to provide it them into the supporting material of HPLC as well as of the UV. Okay, in this way, please do not try to hide the data in hope of saving it for later paper or the another research paper what we want to publish. You may lose the evidence to reinforce your conclusion for this particular paper. Please remember. Okay, if data are too much abundant, you can use supplementary material. I would also advise you to use subheadings to keep the results of the same type together. Okay, and I'm seeing, okay, subheading together, which is easier to review and read. Okay, numbering of these subsections should be for the convenience of internal cross referencing, but always taking into account for publisher's guide for authors. Please keep in mind. For the data, to decide on a logical order, that tells you a clear story and makes it easy to understand. Generally, this will be into the same order as presented into the method section. An important issue is that you must not include the references in this section because you are presenting your results. Okay, and this must be included into the discussion section. तो कोई और रेफरेंस हमें साइट नहीं करना है रिजल्ट में, ये आपके रिजल्ट है। लेकिन जब दूसरों के साथ कंपैरिजन कर रहे हो तो वो डिस्कशन पार्ट में रहेगा, तो वहां पे आप रेफरेंस जो है वो साइट कर सकते हो। नाउ सम स्टैटिस्टिकल रूल्स व्हाट आई हैव एक्सपीरियंस्ड whenever I have come across different manuscript while reviewing or while writing, indicate that the statistical test used with all relevant parameters, for example, mean and standard deviation. 44% plus minus 3%, median and interpercentile range 7 years, 4.5 to 9.5 years. Use mean and standard deviation to report normally distributed data. Use median and interpercentile. For numbers, always preferably you use up to 2 decimal, not like this. Okay, जो एक्सेल में मिला है बच्चा वो उठा के आ जाता है हमारे पास. So this is something which is very, very important for all of us. नेवर यूज परसेंटेज। अभी हम आईपीएल देख रहे हैं तो बोल रहे हैं कि धोनी ने रिटायरमेंट लेना चाहिए या नहीं लेना चाहिए, तो पता चलता है कि 80% लोगों ने बोला कि रिटायरमेंट लेना चाहिए। वो उन्होंने परसेंटेज में दिखाया था, लेकिन वहां पे आंसर दिया था सिर्फ 8 लोगों ने, 10 में से 8 ने बोला था रिटायरमेंट लेना चाहिए और 2 ने बोला था नहीं लेना चाहिए। So when you are talking about small samples, we should avoid presenting it into a percentage way. So rather than you should write it down, 8 out of 10 have said that he should retire. So it is not logical and not scientific to use percentage for a very small samples. Please remember this very, very important. Now, writing down the discussion. Here is something where we all must respond to what the results have mean, and we are supposed to support the result. Okay. Probably to me, I find this is something which is easiest section to write, but the hardest section to get it the right. है ना, उसको शायद लिखना इजी है लेकिन वो सही लिखना उतना ही डिफिकल्ट या हार्ड हो जाता है। दिस इज बिकॉज द मोर सेक्शन ऑफ योर आर्टिकल, हाउ यू गेट चांस टू सेल योर डेटा। आप यहाँ पे अपना डेटा बेच रहे हो, अपना साइटेशन बढ़ा रहे हो। Please take into account that a huge number of manuscripts are rejected because the discussion is weak. If you refer to some of the survey and the data, discussion weak था इसलिए पेपर रिजेक्ट हो गया है। You need to make the discussion corresponding to the result, okay? But do not reiterate the result. Result ही बार बार वही मत दिखाओ आप डिस्कशन में। रिजल्ट में रिजल्ट दिखा दिया है, आप डिस्कशन में भी वापस रिजल्ट दिखा रहे हो, तो ये अच्छी बात नहीं है। Here you need to compare the published result by your colleague with your work using some of the references which you have included into your introduction section. जैसे मैंने introduction section आपको एक published article का यहाँ पे दिखाया था। We should never ignore in the disagreement with our work in turn that we must confront it and convince the reader that you are correct or you are better also. Okay, so this is something which is very, very important. Okay, avoid the statement that go beyond what result can support. Avoid unspecific expression. क्या होते हैं unspecific expression? जैसे अभी मेरा स्टूडेंट एक आर्टिकल मेरे पास लेके आया था कि भाई ऐसे गोल्ड नैनो पार्टिकल हमें बनाना है, तो उन्होंने क्लोरोरिक एसिड को मैग्नेटिक स्टरर पे रखा था और बोला था कि इट वाज केप्ट एट अ हाई टेम्परेचर and it was stirred at a lower RPM. So these are some of the terms which are very unspecific expressions. So good publishers will not support you for it. So quantitative descriptions are always preferred. For example, 35 degrees Celsius, 0.5 percentage, P is less than 0.001. Okay, so it is 0.1 percentage level of significance. We should avoid sudden introduction of new terms or ideas into the discussion section. You must present everything in the introduction and that has to be confronted with your results over here. So this is something what I would advise you to put over here. Okay, and speculations or something on possible interpretations are allowed. Okay, we can do the speculations into the discussion sections about the results that we have produced, but these speculations should be rooted into the fact, into the empirical evidence rather than your assumptions and imaginations. Okay, so to achieve a good interpretation, let us think that how these results relate to original questions or objectives outlined in the introduction section. Do the data support your hypothesis? Ask yourself, are your results consistent with what other investigators have reported? Discuss the weakness and discrepancies of your results. If your results were unexplained, try to explain your result. Is there another way to interpret your result? What further research would be necessary to answer the questions raised by your result? Explain what is new without exaggerating. So this is something what I would like to highlight over here whenever you start your step number 4 to write down the discussion. So what was your step number 1? Step number 1 was to compile the graph and the table. Step number 2 was about writing down the experimental methods. Step number 3 was writing down the result. Step number 4 was writing down the discussion. Is it okay, everyone? This is how will help you to restructure all your entire manuscript. Next is about writing a very clear conclusion. Now, this is the part of the manuscript which will show how our work is advancing in the field from present state of knowledge. Okay. In some of the journal, the conclusion can be a separate section. In some of the journal, it can be the last paragraph of the discussion section. Whatever may be the case where you are submitting your journal, without a clear conclusion section, the reviewer, the reader will find it difficult to judge your work, whether it merits for publication in his or her journal. Please remember the third point on my slide. Okay, विदाउट अ क्लियर कंक्लूजन ये बहुत डिफिकल्ट हो जाएगा कि ये जर्नल के मेरिट में हो रहा है या नहीं हो रहा है. ओके, सो प्लीज अंडरस्टैंड दिस. ओके, अ कॉमन एरर फॉर कंक्लूजन दैट रिपीटिंग द एब्स्ट्रैक्ट. आप एब्स्ट्रैक्ट ही रिपीट कर रहे हो या यू आर जस्ट लिस्टिंग द एक्सपेरिमेंटल रिजल्ट. प्लीज बी अवे फ्रॉम इट. Unimportant, non-significant statement of your result are unacceptable in the conclusion section. Please provide a very clear scientific justification for your work in this section. Indicate the uses and extension wherever appropriate. You can suggest the future experiment if appropriate. Okay, you can go for clinical trial. You can go for preclinical work, kind of a study underway. You can propose a present global specific conclusion in relation to the objective included into our introduction or what we are going to write. The next step is very, very important, is writing down the introduction. Okay, so it should be as compelling as possible, as appealing as possible, because this is the opportunity where you will try to convince your reader that you know that your work is of great potential and it is very, very useful. A good introduction should answer these questions something. Okay, where what is the problem being solved and all that we have discussed earlier. Okay, but editors what would like to see is that you have provided a very prospective consistent with the nature of the journal. Editors पहले वो देखना चाहेंगे कि नेचर ऑफ द जर्नल क्या है, उसका थीम क्या है, उसके प्रोस्पेक्टिव कंसिस्टेंट कंटेंट है कि नहीं वो देखेंगे. सो वी as author need to introduce the main scientific publication on which our work is based, citing a couple of more original important work, including recent review article. Okay, but it may happen, however, that editors also hate improper citation of too many references which are not relevant to your work or which are inappropriate judgment on your own achievement. So they will think that you have no sense of purpose for publishing this article. So please be very clear whenever you are writing the introduction part. Now, as never use more words than necessary, be concise and to the point. Don't make this section into a history lesson. Long introduction puts the reader off. Okay. We all know that we are keen to present our new data, but do not forget that you need to give the whole picture at the first, that is background. It must be original from the global to the particular, as we started with general to the particular problem, guiding the reader to your objective. Hypothesis and objective must be clearly remarked at the end of the introduction. This is something what I also feel, that we are writing novel and first time and first ever and paradigm changing. This is something we should keep it away. Use them very rarely, very sparingly. Okay, because this is, I personally feel, is that exaggerated or an overclaim in the introduction section. Okay, now continuing with it. To confirm this assumption, for example, I am giving you some of the examples of writing down the introduction part. To confirm this assumption, we studied the effect of range of inhibitor of connection challenge, such as this. During controlled experiment, we investigated the influence of HMP boundary conditions. To tackle this problem, we developed a new software verification technique called as oblivious hashing, which calculate the hash values. These all three examples I have taken from the introduction portion of different published paper. So this is also something which is very, very important. Now, these are some of the action verb, okay, that expresses the research actions. Okay, we designed a series of experiment. We determined the complete nucleotide sequence of. We evaluated the efficacy and biocompatibility of a polymer. We implemented a genetic algorithm. Okay, we measured the concentration of cadmium in the lake water surrounding Vastrapur in Ahmedabad, or whatever being there. Okay, so this is something which is very, very important. Next, very important is that when you write down in introduction about your objectivity or about your paper, this paper clarifies the role of so and so. This paper presents, this paper discusses on the theory behind something what you have already seen on the previous to previous slide. Okay, so this is one way of writing down it, of expressing the communication, the verbs. So this paper offers, this paper discusses, this paper details, describes, clarifies, proposes, reports, and summarizes. I am very sure you all people have been using AI very exhaustively. So that is also maybe helping you, maybe not helping you, will lead to a plagiarism as well. Okay. And also other writing software like Grammarly and Quillbolt and WriteMe. So they have been also proposing us with various communication actions as well. Okay. Now, the next very important step is the writing the abstract. Now, abstract is something what tells the prospective readers what you did and what the important findings in your research were. Together with the title, it is the advertisement of your article, I would say. आपका पूरा आर्टिकल का जो एडवर्टीजमेंट है वो एब्स्ट्रैक्ट और टाइटल ही है. ओके, तो मेक इट इंटरेस्टिंग, मेक इट कैची, इजीली अंडरस्टूड विदाउट रीडिंग द होल आर्टिकल. Let us avoid the jargon. Let us avoid the uncommon abbreviations and references into the abstract. Let us be very accurate using the words that will convey the precise meaning of our research. Abstract is something what I have already said on the earlier in my discussion. Short description of perspectives and the purpose of our research or our paper. It will give you the key result and it will minimize the experimental details. Please remember, minimize the experimental detail. It is also very important for all of us to remind that abstract offers a very short description of the interpretation and conclusion in the last sentence. Okay, so if I would have been asked, then in the two way, what has been done and what are the main finding should be there in our abstract. There are online tool also available for preparing the abstract. I'm very sure you all people must be very used to through it. Okay, and next very important is that step number 8 is compose a very concise and a descriptive title. Okay, so opportunity to attract the reader's attention. रीडर्स आर द एडिटर्स एंड द रेफरीज. पहले रीडर कौन है आपके आर्टिकल है? आई होप ऑल ऑफ यू विल अग्री विथ मी. कौन है सबसे पहले रीडर आपके मैन्युस्क्रिप्ट के? एडिटर. और दूसरे रीडर कौन है? रिव्यूअर, रेफरीज. सो दिस इज एन अपॉर्चुनिटी टू अट्रैक्ट द रीडर्स अटेंशन. So let us be concise and descriptive in our title. So, and then readers are the potential authors who will cite your article, है ना, जो वो पढ़ के आपको साइट करेंगे, वो आपका आर्टिकल पढ़ के अपना प्रोजेक्ट करेंगे. So the first impression is very powerful. Okay, so for example, I am giving you one or two example over here. Okay, action of antibiotic on bacteria. Okay, inhibition of growth of mycobacterium tuberculosis by streptomycin. Comment क्या आया इनको? Title should be specific. Think about how will I search for this piece of information when you design the title. Okay. So this is the comment, original comment from the editor. Okay. Now, few more example. You can see over here about the title. Fabrication of carbon rods coaxial nanofiber displaying optical and electrical properties via electrospinning carbon. Revised title, electrospinning of carbon coaxial nanofiber with optical and electrical property. सीधा एडिटोरियल ने कमेंट दे दिया है, इंग्लिश बहुत पुअर है, यू नीड टू हेल्प विथ इंग्लिश, एंड इंग्लिश इज नॉट अवर नेटिव लैंग्वेज, इट इज नॉट अवर मदर टंग. ओके, द टाइटल इज वेरी नॉनसेंस, so very harsh comment received from the editorial. All materials have properties of all varieties, so you could examine even my hair for its electrical and optical properties. So this was something even I found it very harsh. So the authors were asked to be very specific. So I haven't read the paper, but I suspect there is something special about this property. Otherwise, why should you be reporting them? Okay, so that was the editor in chief. I would be running. Okay, so title is something which is very, very important, and which we all should be taking care of it. Keyword, okay, indexing word, they are the label of our manuscript. Okay, it is true that now that there are less keywords are not even being used by some journal, because you can search the whole text into the different search engine. But when looking for keyword, avoid words with a broad meaning and words already included into the title. This is also something which is very interesting, what I personally feel, but still it is very difficult to implement the same. Some just require that keywords are not those words from the journal name, because it implicit that the topic is that only. For example, I come across something whereby I was trying to connect with certain antimicrobial activity of herbal product. So there is a journal of soil biology and biochemistry, and the article are also a keyword as soil. So which they expect that soil should not be there as a keyword. Only abbreviations which are firmly established in the field are eligible to be put up as a keyword. Okay, avoiding those words which are not broadly used. Okay, you need to check the guidelines for authors and look at the number of keywords which are admitted or labeled definitions or range or other special interest. So you are supposed to check the guidelines to the authors as well. Okay. Next, very important is write the acknowledgement. I hope it is self-explanatory. You are supposed to express your sense of gratitude and thank the people who all have contributed to the manuscript, but not to the extent where that would justify the authorship. ये भी ध्यान रखना, नहीं तो लगे एडिटर को कि भाई ये इतना थैंक कह रहा है तो उसको ऑथर क्यों नहीं बनाया है उसने. So this is something all of us should take into consideration. ओके, सो यू कैन इंक्लूड, पूछते हैं कि किसको थैंक करना है, तो यू कैन इंक्लूड टेक्निकल हेल्प and assistantship with maybe some of the grammatical corrections or writing or proofreading. Okay, and you can also thank your funding agencies. You can thank your leadership at the institute for providing you the facilities. You can also thank someone where you have outsourced your sample. Okay, maybe for the project you are supposed to write down the project number or reference number also. Okay, so because it is required for the funding agency, so acknowledgement will have all such stuff being there. The next something where we are creating lot many mistakes is the reference section, the other part of the main part of the manuscript. It is too what we personally feel is something is very annoying, but right now with the availability of reference management software, okay, this is something becoming very simpler and easier for us as well, because otherwise it causes a great headache among the authors, among the editors. Okay, but nowadays it is the responsibility of the authors only. Okay, so there are many tools which are available, and I'm very sure you might be knowing about those Manly or EndNote and Zotero. So these are some of the tools which can be used to manage your reference. Okay. What I would advise at the step 11 is that in text, you must cite all scientific publications on which your work is based. I am repeating. You must cite all the scientific publications on which your work is based upon. But do not overinflate the manuscript with too many references. Please overinflate your manuscript with so many references. Okay. और also avoid excessive self citation. अभी ये फैशन हो गया है सेल्फ साइटेशन का, अपना ही अपना को कॉपी करना. ओके. सो दैट आल्सो शुड बी अवॉइडेड. साइटेशन एंड द पब्लिकेशन ऑफ द सेम रीजन, तो अब नया फैशन शुरू हो गया है कि चलो मेरा वर्क नहीं साइट करूंगा, तो I will cite my colleague's work who is in chemistry department, my colleague's work who is in pharmacognosy department, my colleague's work who is in my neighboring college. तो पर्टिकुलर रीजन में से भी आप साइट करना थोड़ा अवॉइड कर दीजिए, बिकॉज दिस इज ऑल समथिंग विच हैज बीन noted down by the editor. Okay. So there are authoritative information on journals policy on citation when you will refer to the journal's guide for the authors or author's guideline. Okay. This is something what I had to say regarding the references. And at the end, what I finally feel is check the spelling of the author's name, affiliations, year of the publications, what you have cited, usage of ATL or something, or the syntax, punctuations, whether all references are included or not. Okay. So this is something what I feel is very, very important for all of us to take into consideration whenever we write an effective article. Now, to summarize and to sum up before I take up on your questions. So this is something as a whole we have taken up. Okay. So title, citing drug, intervention, context, design, and main finding. Use subtitle sparingly for study names. Okay. Abstract, which should be understandable, comprehensible by itself, introduction, method, result, conclusion. What should be there in introduction? What should be there in method, result, discussion, and reference. Okay. So this is something I had, and I'm very glad to share with you all. And I'm very much sure that this has added a value to what you were already knowing it, and you already are publishing it, your work. Okay. So all the content of the slides were being taken from the different references. When we are talking about effective scientific research paper, I should not fail to cite the references from where I had taken all these steps. Okay. So a step-by-step guide for beginners. This is a very good paper. Again, there is a very good writing for books and journals. So you have the entire elsewhere database available. Then this is also practical 6-step approach. Okay. Then you have something from the Purdue University, entire guidance document is there. You have research academy of elsewhere, which gives you an interactive course on writing an effective paper, writing abstract, writing material method section, and it is free available. So you all of us can enroll into it. Coursera is also offering you the free online course for writing an effective presentation, writing an effective research paper. The first article which I discussed is of my MPharm student, and this is also something of our MPharm student. And the third one, which I would have discussed depending upon the time available, but it's already 12:10. Otherwise, the first and the third article being mentioned over here were first filed for patent have been published, and then we sent for publication. So what Moria sir was describing in his first lecture, that patenting was done and then later on it has been published, and that has been the work of MPharm students. Over here we had in this article only eye irritation study, Dre's eye irritation study into the rabbit. Over here, we had only ex vivo permeation study into cornea, but we could successfully publish it into. This was out of our financial grant we obtained from UAE. Thankful once again to the organizer, Sardar Patel College of Pharmacy, and especially to my good friend Dr. Samresh for inviting me over here and to share something on how to write an effective research paper in a very conventional and a way whereby with our own human intelligence we can do a lot without using the artificial intelligence. But I am a very advocate of using artificial intelligence wherever it tries to reduce our efforts. So thank you very much. You can see over here a difference in the H index of Google and Scopus. So this is whereby self-citation have been included. Over here, the self-citation is not included. So this difference should be not there. It should be as minimum as possible. Okay. So thank you very much. I am happy to take the questions wherever being there. Over to you, organizers. If, yeah. Yes, Gunjan madam, you are mute.
Thank you so much, sir, for the insightful and informative session. May I request all the participants, if anybody have any questions regarding the session, you may ask to the Ketan sir. And please raise your hand.
Yeah, thank you, Ketan sir. It's really very insightful session. I found each and every topic you
Thank
have
you. Thank
nicely
you.
explained. So as Gunjan madam said, if anyone is having any question, please you can raise your hand. We'll keep you on mute so that you can directly ask the question to Ketan sir. Plus, sir, please you can unshare your slide.
Okay, just hold me.
Okay.
I have
Or you can
stopped
ask.
sharing my slide.
Okay, okay, fine. So there's an option called raise hand. You can just click there and I'll give you a chance to unmute so that you can directly ask the question. Or else you can put your questions in the chat box. If you want, you can also directly put your questions in the chat box. Ketan sir, also you can see in the chat box. I can see there are lots of
Okay.
overwhelming messages, very informative,
Thank you.
very
Thank you.
fruitful session. I believe all the audience get something new and very, very important information from this session. Yeah. You can also write in the chat box if you want to ask in the question directly to Ketan sir. There's a question I can see, but it is all about from the review article, I think. Is there any difference between
Yeah.
systematic
There is a
review?
question
Kajal Ghoshal.
regarding patent work is published, then how is it possible to get the credit for patent since the material is available openly? First, we have to ensure that our patent has been published and filed successfully. Then you only communicate your manuscript to the publisher. Okay. Reference management software mainly is open access, freely available. Okay, and not is a paid one. Okay, so these are some of the, even we have software reference management available in the MS Office as well, so which also you can use it. Pre-journal for pharmacology, like on that particular day, Moria sir has shown us one slide about open access and subscription mode. So the open access mode has been more prevalent nowadays since the COVID era. Otherwise, all of the publications were made onto the subscription mode. whereby authors do not have to pay the fees for publication. So most of the papers or most of the journals, what you would see, would accept you with the fee publication. But please ensure that the publications is made into the peer-reviewed journals, which are listed either in Scopus database or in Web of Science database. Okay. Otherwise, the other journals do not have a very systematic and a scientific peer review system. And if even if we refer those articles, we would be misled in our conducting the research. Sir, I would like to know that is it correct and ethical to prefer ex vivo study, that is using cell line instead of in vivo study, to prove the objective of our work? Yes, very fine. It would be very much ethical to have ex vivo study in terms of cell line, or maybe study doing the excised tissue. But at the same time, we know the performance of the delivery system or the dosage form would show variation when actually it goes into the system. Okay. So biological system. So maybe the reviewers would be interested in your preclinical or clinical study, but that also depend upon the journal theme, objective, and the outreach. Otherwise, both the articles what we have shown have the ex vivo or the cell line study, because we do understand it would be very difficult for a pharmaceutical student to go with the in vivo studies also within the 6 months or 10 months of their MPharm dissertation. Discussion is very important. Guide me for the length and depth I should prepare for achieving the selection in reputed peer review journals. I'll pass on you the entire points which I have gathered through my experience of writing down discussion. Okay, which is in fact very, very important, whereby you discuss the trend and the association that you have observed in your result, and you compare and contrast, you write down implications of your study, the conclusions of your study, which is being seen or being importance with the peer reviewed journals. Otherwise, you go on reiterating the same thing what you have written in the result, is something will ask for a rejection from the journal. Another question, what I think, I hope I have covered patent also, reference management. Fine, but as I said, the take home message जो हम कहते हैं कि किताबों से पढ़ के या लेक्चर पढ़ के हम तैरना नहीं सीखते हैं, ना ड्राइविंग सीखते हैं हम, तो वैसा ही है कि मैन्युस्क्रिप्ट का राइटिंग या जो है वो ऐसे पढ़ाई करने से या लेक्चर अटेंड करने से एक आपको स्पार्क या आपको एक इंस्पिरेशन या एक गाइडेंस दे सकता है। लेकिन जब तक आप लिखना नहीं शुरू करोगे, पढ़ना नहीं शुरू करोगे, तो वो मुश्किल है कि वी वुड बी एबल टू give up a draft of our manuscript which can be accepted. And for that, you need to read at least minimum 5 research papers, original research article, which is related to your work. So that will be like torch bearer or lighting for your effective research paper writing. How can we improve citation for our research? We can improve the citation our research by, I would say, putting it into the social media. I would that is one particular aspect of, or by disseminating at various platform, you can improve the citation of our research. Okay, so this is one particular way of improving the citation. Okay. So in an age of internet, I hope citations are going to improve for all of us.
Yes, sir. So
Any
it's
more questions? I would be.
I think we are
I
running
think
out of
someone is
time.
already. Okay, it is 12:20.
Yeah.
Future projections will come under discussion, or it will be under conclusion. It will be under conclusion. Like it can be further substantiated with the preclinical study in large animals, or it can be substantiated with the clinical study, or it can be so future projections would be under the conclusion. And conclusion is the last paragraph of your discussion part. Generally, many of the journals don't have the conclusion as a separate section apart from the discussion.
Sir, am I audible?
Yes, yes, you are audible.
Thank you so much, sir, for this insightful and informative session. Your knowledge really helps and deliver made the topic clear and engaging. I truly appreciate your time and effort put in sharing your expertise. So now, from the behalf of Sardar Patel College of Pharmacy and from all the participants, I appreciate you. And sir, please accept the certificate of appreciation from the Sardar Patel College of Pharmacy, Gujarat Technological University, as a token of appreciation and gratitude, recognition your invaluable contribution. Now, participants, I request all the participants,
Thank
please
you.
kindly fill the feedback link.
Thank you very much for your appreciation with the certificate. I humbly accept the certificate of appreciation. And I also wish you a very fruitful publishing to all the participants. So, as when I started on a note that the real outcome of this FDP would be when we come up with the first draft of our manuscript.
Sir. Thank you, sir. So, sir, just hold one
Thank you
minute.
so much.
I just want to
Yes.
request all the participants kindly please turn on your camera so that I can take some screenshot. I request all the participants who are available right now kindly turn on your camera. I request again all the participants please turn on your camera. I like to take some screenshot along with your speaker today.
I request organizers also to share the feedback of the session with me so that I can improvise the content as the delivery of the content of the topic.
Yeah, sure, sir. Sure. We'll definitely share the
Yeah,
same with you.
many people, I think some of them have raised their.
Okay. I just, KM Shiva, would you like to say something? Any feedback?
Someone has raised
KM.
the hand.
Yeah, yes, yes, I can see.
I will post my mail ID. I am serving as associate professor at the first independent pharmacy institute of the nation, LM College of Pharmacy, Ahmedabad, established in 1947.
So day 3 feedback form is already shared
Fine,
in the
sir.
chat box. The
Okay.
participant can now share your valuable feedback in this Google form.
Okay, sir.
Thank you all.
Okay. Thank you, Ketan sir, again, and all the participants to
Thank you,
join.
Samrit sir.
Thank you, Ketan sir, again
Thank
to
you.
accept our invitation and to deliver
Thank
this
you. Thank you.
insightful session with us. Thank you, sir. Now, audience, please
Thank you.
kindly fill this feedback form and then you can leave.